Hello, my name is Lars Westblade and I'm from Weill Cornell Medicine. And in this video, I'd like to tell you about Staphylococcus aureus beta-lactam resistance mechanisms. In order to do that, I have to tell you about the target of beta-lactams, and that is the bacterial cell wall, which is also known as peptidoglycan. The bacterial cell wall is cross-linked together by enzymes that are known as transpeptidases. Transpeptidases are the specific target of beta-lactams. And for that reason, they are also known as penicillin binding proteins. Beta-lactams bind to and irreversibly inhibit penicillin binding proteins such that the cell wall cannot be synthesized. Inhibition of cell wall synthesis results in cell lysis and death. In Staphylococcus aureus, when we want to determine beta-lactam susceptibility, we test a number of different beta-lactams. We test penicillin because that predicts susceptibility to penicillinase LABR penicillins. We test oxacillin and sulfoxidin because they predict susceptibility to all beta-lactams, with the exception of ceftaroline. In order to determine ceftaroline susceptibility, we test ceftaroline itself. And just to remind you, ceftaroline is an anti-methicillin-resistant Staph aureus cephalosporin. So methicillin resistance is the most important mechanism of beta-lactam resistance in Staph aureus. And it's due to the acquisition of an alternative transpeptidase called penicillin binding protein 2A, which is encoded by MEK-A. PBP2A can cross-link the staphylococcal cell wall in the presence of beta-lactams, and that's because beta-lactams have a very low affinity for penicillin binding protein 2A. And to remind you, penicillin binding protein 2A confers resistance to all beta-lactams, including penicillin, with the exception of the anti-MRSA cephalosporin ceftaroline. If a Staphylococcus aureus isolate does not encode MEK-A and therefore does not produce penicillin binding protein 2A, we call the isolate methicillin susceptible Staph aureus, or MSSA. If the Staph aureus isolate does encode MEK-A and does produce penicillin binding protein 2A, we call it methicillin resistant Staph aureus. And both oxacillin or sulfoxidin can be used to assess methicillin susceptibility in Staph aureus. I'm showing you a photograph of a physical model of penicillin binding protein 2A. Highlighted in blue is the transpeptidase domain of the enzyme, and that's the business end of the enzyme. And highlighted in orange is the non-penicillin binding domain. This model was generated in collaboration with a wonderful group of high school students from Hostas Lincoln Academy in the South Bronx. So I want to review the resistance profile that we might expect for methicillin-resistant Staph aureus, otherwise known as MRSA. The mechanism, as we discussed, is due to the acquisition of penicillin binding protein 2A. The strain designation would be MRSA, and the isolate would be penicillin resistant, oxacillin resistant, sulfoxidin resistant, and ceftaroline susceptible. There is another variant of penicillin binding protein 2A called penicillin binding protein 2C, which is encoded by MEK-C, and it confers a very similar phenotype to penicillin binding protein 2A. But often the isolates are sulfoxidin resistant, oxacillin susceptible. And importantly, tests that are specifically designed to detect MEK-A or PBP2A will not detect MEK-C or PBP2C. MRSA isolates can become resistant to the anti-MRSA cephalosporin ceftaroline, and they do so by generating mutations in the PBP2A protein. In that situation, there will be resistance to all of the beta-lactams that we typically test against Staph aureus. Penicillin resistance. Penicillin is an excellent agent for treating methicillin-susceptible Staph aureus infections, and resistance is conferred by the production of a penicillinase called BLA-Z. BLA-Z confers resistance to these penicillinase-labeled penicillins, but not to other beta-lactams. Historically, very high rates of penicillin resistance among Staph aureus isolates resulted in decreased use of penicillin. However, that trend seems to be changing, and we're seeing increased susceptibility to penicillin in Staph aureus, particularly in MSSA. So there's renewed interest in using penicillin to detect methicillin-susceptible Staph aureus infections. And penicillinases work by hydrolyzing the beta-lactam ring of penicillin, rendering the molecule inactive. So when we consider penicillin resistance due to BLA-Z production, we can designate the strain penicillin-resistant Staph aureus, we're resistant to penicillin, and we're susceptible to oxacillin, sulfoxidin, ceftaroline. There is this curious phenotype that we can see called borderline oxacillin-resistant Staph aureus, or BORSA, where we have hyperproduction of BLA-Z that results in resistance to penicillin and oxacillin, but susceptibility to sulfoxidin or ceftaroline. We also have another very interesting phenotype, which is rather rare, but it can be observed, called modified oxacillin-resistant Staph aureus, and that's arises due to mutations in the native penicillin binding proteins associated with Staph aureus. Susceptibility profile is penicillin resistant, oxacillin has a variable result, cefoxitin is resistant, ceftaroline is susceptible. On this last slide, I've summarized the resistance mechanisms that I've discussed. Thank you so much.